So welcome everyone to our webinar today. We have a really interesting topic on the infrared imaging used as a new medical diagnostic or screening tool. Before we start with the actual topic, we always like to poll our audience a little bit. So I have a little poll here, just asking you how you would rate your knowledge of infrared imaging. So that should appear on your screen right now. You can just click on your answer choice, depending if you are interested, you don't have any experience with this, or if you even consider yourself an infrared imaging expert, then just let us know. It helps us sort of understand a little bit more of the audience that we're talking to. I'll give it another 10-15 seconds. Okay, 5 seconds. There's more and more answers coming in. All right, I think we can call it. Great. So that's very interesting. A good mix of people in your audience. Even some people calling themselves infrared imaging experts. So that's great. Hopefully, we'll get some very interesting questions at the end of the webinar. So here we are. Let me just switch to my screen here. So today, we are talking about medical imaging. I'll do a quick introduction talking about Telops if you're not familiar with us. Exosense as a whole. I'll talk about our product line. Won't spend too much time on that. I'll talk about the medical imaging potential for infrared imaging and just a few quick examples. But of course, our main presentation will be handled by Professor Ahmet Koluman and his PhD student, Batuhan Akilli, who'll be talking about measurements that we took together in Turkey earlier this year. Some very interesting results. We'll end the session with some questions and answers. So during the presentation, feel free to write down your questions. We'll be able to take them in at the end. You don't have to wait for the very end to write down your questions. So it's going to be in the GoToWebinar platform on the right. There's a question section. You can click in there and it's open to you. But just a quick note on Exosense and Telops. But Telops was founded in 2000 in Quebec City, which is where I am right now. So we now employ about 100 employees from various backgrounds and scientific degrees, PhDs, master's engineering degrees. So we're really proud of our R&D and how we've been developing IR imaging in the last 25 years. And we're really established as we're leaders as thermal infrared imaging solutions. Now, a couple of years ago, Exosense, the Exosense group, bought Telops, so we joined a really interesting group of electro-optics companies. So that's really exciting. It's opening a few doors into collaborations and new opportunities for what we can do, what kind of technology and science can be accomplished with our instruments. So we do offer a full suite of infrared imaging cameras as well as hyperspectral systems, and we even have an aerial methane leak detection and quantification system. So quick glance at our product line. We have hyperspectral systems that I mentioned here, multispectral, full scientific camera, high dynamic range camera, the fastest IR cameras in the world as well. Today we'll be focusing on this little guy here, the Radia V60. So a new entry-level camera. We say entry-level because, of course, they're cheaper than the other high-grade, super fast cameras, but they're still scientific grade instruments, and they have a really interesting high-level data quality that you can acquire with them. We do a lot of instruments, and therefore we cover a lot of applications here. I don't even have medical application on my slide here because we cover so many things, and medical research is just starting to really look into IR imaging. So that's why I'm so excited that we're talking about this today. I think there's a lot of potential and growth here that could be happening in the medical imaging world. So I mentioned the Radia V60. The measurements we'll be talking about today are done with the V60. So it's a long wave camera, an uncooled microbolometer camera. So 640 by 480 pixels that can go up to 60 hertz. Yes. It's not aimed towards high-end experts that need to do extremely advanced types of IR measurements, but industrial R&D, university laboratory, medical research facilities. It's a great tool for this, and it offers a flexibility that you don't necessarily have compared to other similar cameras in the market. Being focused manually, being able to swap lenses, and that low swap packaging, so very small cameras, still offering scientific grade quality measurements. That's really, really interesting for research work. So a few examples of the kind of images you can get with this, including just my son here at the bottom. I just happened to have a fever a few months ago. The kind of scientific dad that I am, just testing some instrument on my son. But you can see the camera is very, very small. It holds in the palm of your hand here. When we talk about medical imaging, thermal imaging is typically handled with long wave, in the long wave band of the infrared spectrum. It's useful for higher sensitivity to detection at lower temperature. So medical imaging typically remains at low temperature compared to other types of infrared imaging. So that signal with long wave will come very much near the surface level. So there's a strong limitation of absorption through body tissues. So you can't image something deep inside the body with long wave imaging. It can be done with broadband cameras, but you can even do multispectral imaging as well if needed, using spectral signatures depending on what kind of imaging you do. But for medical screening and diagnostics, there's been more and more research has been done for decades, but there's really good overview papers. There's a good one that I used here as a reference, just describing the kind of issues and diseases that can be potentially screened and diagnosed with IR imaging. Of course, IR imaging is not the only tool you need in your arsenal for a good medical diagnostic, but it's a really interesting, completely passive tool that can help the final diagnostic. So there's a lot done on breast cancer, being near the surface. This is really useful. We'll be talking about that today with the main story. But there's plenty of others here that I won't go through the whole list, but you can see the kind of examples that are possible to image with infrared imaging. It's a really, really interesting work that's really just starting in the last few years, last decade at maximum. So I'll show you a few examples that were taken with Telops cameras. One great example here at the University of Sherbrooke in medical research. They were looking at cancer clusters, so how to look at cancer clusters, spread, metabolic rate in a biological tissue. So they were inserting cancer cells of different kinds in chicken embryos and then doing some imaging in there. And you can see those cancer cells are spreading quite easily. So a really interesting work happening there. For human bodies, we can see here a great example of vascularity imaging. So that can be great for blood clots, vascularity issues of any kind. You can see those veins very, very well using long wave imaging like this. Eye issues is a great example. Sorry for the slightly creepy video here. But there's so many measurements and diagnostic or screenings that can be done for eyes with thermal imaging. So we have dry eye, diabetic retinopathy, glaucoma, conjunctivitis, inflammatory diseases. Inflammation is a typical good indication of different diseases, and because it's inflammatory, you have a difference in the thermal signal coming from the body tissue, not just for the eye, but anywhere on the body. For fever detection as a whole, as a passive remote system, The tear ducts, so just around this here, around the tear ducts, is a great area to have a fairly reliable measurement for the temperature of the body for fever. So really interesting work. Again, just starting in this field. I wanted to mention microscopy imaging as well. This is interesting, not necessarily for direct remote sensing diagnostic, but for as a tool for sample analysis or medical tool material research that can be really useful. We can see here on the right just distal cells, so we can see the cellular structure in a housefly. So really, really magnified image here, still in infrared imaging for thermal response. But now, enough about me, enough about Telops story. I want to talk really about our story, our main story. So I'll let Professor Ahmet Koluman and Batuhan talk about what they did, what kind of result they took, and I'll introduce them briefly here. So Batuhan is a PhD candidate in biomedical engineering. He has a master's thesis focusing on hypochlorous acid application in wound healing. He's currently a lecturer at Afyon Kocatepe University in the Department of Biomedical Technologies. And Professor Ahmet Koluman is a professor in biomedical engineering, specializing in rapid diagnostic and detection systems, as well as telemedicine at Pamukkale University. So I'll let Professor Koluman share his presentation. I'll stop sharing mine. Here we go. So the scene is yours, Professor Koluman.
Good morning or good evening, wherever you are. In these few minutes, we will talk about our preliminary study about using thermography for detection of many pathogenesis and many different unpsychological positions. To start with, general clinical use of infrared thermography, it is mainly used in vascular disorders, musculoskeletal assessment, neuropathic conditions, and oncology adjunct imaging. It is supporting early detection of temperature asymmetries related to the inflammation or circulatory impairment. It also provides non-contact. It is really important for us that is radiation-free and screening test which can be repeatable and repeatable for trustable results. FDA also approved the use of infrared thermography systems. Most medical grade infrared thermography systems require FDA 511K clearance before clinical use. FDA evaluates the safety, accuracy, substantial equivalence to previously approved systems. It is typically approved as an adjunctive imaging tool, not a standalone diagnostic method, which is the main idea we started to work on it, because we need a screening method to see if anything happens on the body can be detected using infrared cameras. The purpose of the study is to objectively evaluate the body surface temperature distribution of patients from different clinical diagnostic groups using infrared thermography to standardize the imaging conditions and to investigate the application opportunities of this method as an adjunctive imaging tool in clinical diagnostic workflows. We also aim to optimize the patient preparation, room condition, device setup, imaging protocols, and to see the repeatability, accuracy, and clinical usability of the measurement technique. So as I'm going on, I will leave the floor to Batuhan. He will talk about technical details of the method. The floor is yours, Batuhan.
Hello everyone, have a good day.
determination of patients, we identify patients group based on literature reviews and research. We select four different disease group. As you can see, these groups are breast cancer, juvenile idiopathic arthritis, peripheral artery disease, and thyroid disease. These patients are currently under treatment by doctors at Pamukkale University Hospital. We also form a healthy group. Both patient and healthy groups individuals participate in the study voluntarily. Next page. Preparations of the room. We use a standard room in the hospital for infrared thermal imaging. This room is approximately 20 square meters. It is very important to keep the room temperature and humidity levels constant. Sunlight can make errors thermography image. Therefore, we close the windows and the curtains to block sunlight for both these imaging errors and constant temperature levels. We also use the air conditioner to stabilize the room temperature. However, turn off the air conditioner during the actual imaging to avoid errors. We provide a screen for breast cancer patients to change clothes privately. We also prepare gowns for them to wear before imaging. Next page. Setup of the camera. The Telops Radia V60 device is small, compact, and portable. Due to this advantage, we place this camera on the table using a small tripod. It is easy to set up. The camera have two cables, one power cable and one connect cable. We connect the camera to a computer, operate it. We take the image using the camera's computer applications. We capture 150 frames at a time. All raw image data is saved on the computer storage. Next page. Conducting thermography shoots. Firstly, the patients and the healthy groups sign ethics committee approval forms. We take thermography images of patients with a clear diagnosis and healthy individuals. Everyone who participated in this study rests before the imaging process. We wait their body temperatures to return normal levels. The imaging process takes about 15 seconds on average. For some groups, we take thermography images in different positions. The camera is at Pamukkale University Hospitals for only 3 days, from May 26 to May 28, 2025. During this time, doctors help us identify the voluntary patients and healthy individuals for study. Professor Ahmet Koluman, I leave the floor to you again.
Okay. During these 3 days, each patient was called to the imaging room one by one, separately, and imaging procedure included only one member of the technical staff and one medical doctor, and each patient was explained in detail for, especially for the pediatric patients, and even more detailed and age-appropriate explanation was provided. Patients were allowed to rest before the imaging as we made static imaging. We didn't do any dynamic imaging. Patients were allowed to rest before imaging and any perspiration was wiped off to ensure proper preparation. Afterwards, static imaging was performed. We didn't perform any dynamic imaging due to we need to understand the limits of the static imaging. For breast cancer, it is a popular cancer type, as you know, and we choose mostly the post-operative patients. And in this thermographic images, we have 12 individuals in the patient group. This is the patient group's images. So as we focus on the patient group, for the first picture on the left side, you can see on the left breast, there is a widespread vascularization. Also for the right side, you can see the vascularization. This is a post-op patient. As we move forward further to another disease, this is the peripheral artery disease, PAD. We wanted infrared thermal camera to demonstrate the blood flow impairments in the leg vasculature resulting from the stenosis or occlusion, let's say blockage. And we wanted to see if there is a difference between the cold and heat areas. Let's move to the images. Both of them, the patients, these are the patients. You can see the heat changes, and these patients have blood occlusions, blockage on the red areas where the heat increases. As we move to another group, this is also the patient group. This is interesting. This one is especially interesting. You can see the vascularization here. You can see the vascularization here, but you can see the widespread heat distribution on the patient. Why we love thermal camera, I would like to tell here, some patients had really increased pain that they cannot stand alone. So we asked them to sit down, and the areas do not change by sitting. So we had the result that thermal camera can be used by standing or by sitting, which does not affect the result. You can see the heated areas of each patient. Juvenile idiopathic arthritis is another question of concern for the young patients. Thermal alterations surrounding the knee joint were successfully observed in this study. We found that the regions of inflammations are distinctly identified as I move to the group. This is the healthy patient. As Batuhan indicated earlier, it is May, late May in Denizli, where the outside temperature was around 37 degrees. So this is homogeneous distribution, but in this patient, we can see heterogenic distribution around the knee. As we move forward, you can see the distribution here, also heterogenic. But for this image, this patient has a pain on the left leg, on the upper of his knee. He indicated this area. I don't know if you can see my arrow. And this patient showed exactly the area that I am showing now as a pain area. For trach disease, we had a large group of patients, around 54 patients and 39 control group. And we had the cervical neck region conducted infrared thermal camera application. And we found out that in the healthy group, the heat distribution was not so significant, but in the patient group, there was a significant heat distribution in the thyroidic area. It is also the same. This is our health group, and this is the patient group. This patient has pain on mandibular area. As a general evaluate the results, the infrared thermal camera was small. It was handy. It was non-invasive, not invasive, and radiation-free, painless, and rapid. And it constitutes a preliminary screening tool and the method, and it was showing promising results. So as we move forward, we see that it is applicable to many of the diseases of concern, and additional some other diseases. So as a preliminary study, we found out that this system needs validation. The validation should be done in repeatability and reproducibility studies, and these studies also should be done in a controlled area. The verification also needs to be done using a calibration, focus accuracy, and noise assessment. That will be more, that will increase the comprehension of the system. As we know, as it's a thermal camera, it does not use any medical device standardization method, but as they use to the patients, to the disease control, quality management flows, ISO 13485 principles should be applied, and standard operating procedures should be prepared. And we applied for a project in Turkey to a RECEP project where we want to prepare this clinical use and standardization and quality management of the thermography system of this device. To end with, it is a useful and unique method to control the and screen the pathogenic lesions before they reach to the doctor. It can be a nice approach for a telemedicine. It can be a nice approach for any kind of screening method. I would like to thank your attention, for your participation, and we also appreciate the support of all clinical staff, technical teams, and patients who contributed to study. Thanks for your listening.
Great. Thank you very much, Professor Koluman and Batuhan. I'll invite our attendees to write down any questions in the little chat messenger or the actual question box here on the right. I will mention we had that small technical difficulty with the sound. We'll try to do some sound editing on our end here to smooth out the audio when we're sending out and publishing the webinar video as well. Now, for questions, let's see the questions coming in. I'll start with one of my own. Well, maybe a clarification. You mentioned in your presentation that the raw images were stored in the data bank and all of that. Just to clarify with people that are more familiar with the IR imaging aspect, were you using actual raw images, or there were still temperature images, right? Because in the imaging world, there's a difference between the raw image that the detector gets and the calibrated image that you get, and then you can still store after that. But are you talking about pure raw images, or it's still the temperature measurement that you store after, but you don't really process afterwards?
We are using calibrated images. Batuhan, can you share one image for the listeners to see what we are talking about? We will share just a random image from this study to see for everybody to see that each pixel has its own temperature indication on it. Antoine, I think Batuhan needs a screen sharing.
I just made a presenter, okay. So should be able to. Yes.
Okay.
Perfect.
This is one image. As we go, before it, you can choose the modes, you can choose the colors that you move forward. Batuhan, you can move around the colors. Yes, you can adjust the colors. The software lets you to move around the colors, and as you focus,
What do you mean by focusing?
by focusing, zoom,
Oh, the magnifying. Okay.
magnifying. Yeah, as we magnify, you can see each pixel has an indication on it. Yes, this is what we are talking about.
So yeah, so every pixel in the software will have a temperature.
I don't know if you can see each pixel changes the temperature.
Yeah, you can actually zoom in a little bit more. Even a bit more. There we go. So you have the actual values.
So each image has a numerous data for us to work on it, for image processing and for, let's say, artificial intelligence, machine learning, optimizing. So we use the calibrated data, and we still work on the machine learning part to see the optimization of each disease, which is a huge work. So each pixel has its own temperature signature, let me say signature, that you can move forward. I don't know if it's sufficient for you.
I think that makes it clear. Yeah, it is a calibrated temperature image, and then you're using every pixel as
Yes.
a tool here. That's great. And do you do any averaging of the images of the frames?
No, we haven't done averaging for this webinar, but we started averaging the results as for disease and numerous of patients. We are trying to optimize the averaging details for each patient and versus the control.
Okay. All right. Now I got another question here. Could you please
Yes.
comment whether active thermography will be beneficial for medical IR imaging? Interesting question.
Can you please repeat, Antoine?
Yep. So it's please comment on whether the active thermography will be beneficial for medical IR imaging.
Okay, we have two methods: active or dynamic, static or passive one. For the passive one, if I am at home, I'm talking about telemedicine. I want to screen myself, let's say monthly or bi-monthly. I do not have an option to cool down myself controlled in a controlled environment. So static screening was ideal for telemedicine purpose at home. But if we are talking about hospital conditions, patients should be cooled down approximately 1 or 2 Celsius, and let patient to heat up again. That we know that if any pathogenity occurs, the patient will heat up more quickly. So for the hospital condition, the dynamic will be or active will be ideal, but for the at home telemedicine purpose, I think static and passive one will be ideal.
Great. Something else coming in. Do you see yourself starting? Well, do you see yourself trying this on other diseases, other issues?
In fact, the device, we fell in love with the device, to be honest. It was really handy. It was really quick. It was not traumatizing the patients. We really love to use it in many diseases, and we have lots of ideas in the area of concern. To give one idea, we would like to use the device for the diabetic foot. It will be a great approach for us. We also want to use the device for any changes in the testicular region, let's say varicocele. We also want to use the device for any healing of the wounds could be decubitus or like. So we have lots of idea in the area of concern. So why we applied to a project to have the device to move in the area of concern freely.
Very, very interesting. I I just noticed that your sound just happened to be fine during the question section of the webinar. So not sure again why that technical issue happened during the presentation. Hopefully we'll smooth it out. If there are no other questions, I think we'll be able to end it here. I want to thank again Batuhan and Professor Koluman for the presentation. Really, really interesting work, and more to come. So very exciting stuff. So thank you very much, and I hope you have a great end of your day.
Thank you. Have a nice day.
Thank you.
Have a nice day.
Thank you.
Bye, everyone.